Hello and welcome to Unboxing Biology with Smriti. In today's video, we will be discussing about pleiotropy, right? Pleiotropy, or you can say we are studying about pleiotropic genes. So now, why this term is used, or what does it mean? So it means when the gene is responsible for more than one character, or trait, right? So whenever we have studied previously, so there we have already seen that one gene is responsible for the single character, but here in the case of the pleiotropic genes, the genes are responsible for more than one character, right? So we can get here the example of Drosophila, right? So this is so it's Drosophila, right? So in Drosophila, the gene which is responsible for the size of the wings also affects or influences character like longevity, right? Secondly, structure of reproductive organs, reproductive organs, bristles, right? And reduction in the eggs production or the production of the egg, right? So here we are taking the example of the Drosophila. Here we are seeing that the gene which is responsible for the size of the wings also influences or affects the growth or affects the other characters like longevity, structure of reproductive organs, bristles, and reduction in the egg productions, right? Now, this affection, or you can say this affection rate, is not equal, or it is influencing everything here, every four characters, right, with the wings, size of the wings, but not in equal pattern. It means that it can affect or influence longevity maybe very much, structure maybe somehow less, results again can be very much, or it can be low, right? So here what is I'm trying to say you that Drosophila, the gene which is responsible for the size of the wings, can also influence the longevity, structure of reproductive organs, bristles, and reduction in the egg production, right? But the affection rate is not equal, right? Now we will be taking a second example, which we can understand very easily, and it is sickle cell anemia, right? So second example of the pleiotropic genes or the pleiotropy is sickle cell anemia. Now I will tell you, suppose this is a chromatid I have drawn here, right? To understand pleiotropy more easily, this is chromatid, here is the gene, okay? So the gene which is present on this chromatid affects or influences more than one character or trait. So that's why if this thing is happening, then it is the example of the pleiotropic gene or the pleiotropy, right? Now we'll start with example sickle cell anemia. Sickle cell anemia is actually an autosomal recessive disorder, right? So this is autosomal recessive disorder, and the sickle cell anemia occurs due to the mutation, and the mutation type is point mutation. Okay, so I will be discussing why we're telling here the point mutation thing. So now sickle cell anemia we will be studying as an example of the pleiotropy. So if you know the structure of the hemoglobin, HB, indicated by HB, hemoglobin. Okay, so what happens here in hemoglobin? There are two structures present, two alpha polypeptide chains, okay, which has been numbered as alpha 1 and alpha 2, right? And there are present two beta polypeptide chain, right? Polypeptide chain. Now, here in the case of sickle cell anemia, in the hemoglobin, one of the beta polypeptide chain is affected or is being showing the point mutation thing, right? So we will be discussing here the structure, not structure actually, the sequence in the hemoglobin. In hemoglobin, one of the beta polypeptide chain, point mutation is being shown, right? So we are 1 minute. So this is point mutation, right? Now let us suppose these are the amino acid chains. Obviously this is polypeptide chain, so amino acids will be there, because polypeptide chain is made with the amino acid chains, right? So let us number these. Suppose this is 1, 2, 3, 4, 5 and 6. You can draw one more here if you want. So now this is 7, right? In first, valine is present, valine amino acid, right? Then histidine, then leucine, then thrombin, then prothrombin. Here glutamic acid is present, and again here the glutamic acid is only present, right? So this is the normal sequence of the amino acids which are present in the beta polypeptide chain, right? But in case of this is shown in a normal person, normal in the sense of sickle cell anemia I'm talking here, okay? So normal person shows this sequence, these sequence, okay, right? Now if a person is having sickle cell anemia, so what happens in the sequence? Everything will be same as this only, but let us draw again 1, 2, 3, 4, 5, 6 and 7. If a person is having sickle cell anemia, so the sixth amino acid, which is here glutamic acid, is replaced by valine, right? Now every sequence will be again same: valine, histidine, leucine, thrombin, prothrombin, and but at the sixth position, that was glutamic acid, it will be replaced by valine, okay? And again the seventh will be the same as glutamic acid. So now, because you can say here in sickle cell anemia, two things are happening. First, there is point mutation, right? Point mutation. Second thing is the glutamic acid at the sixth position is being exchanged by valine, right? So now Again, the affection of that thing. Which thing? That gene is replaced by the valin. Now, this affects the two things. First, structure of the hemoglobin. Structure of hemoglobin, right? Because of this, when the structure of the hemoglobin gets affected, it carries less number of oxygen as compared to it was taking up previously, right? So now it is taking less number of oxygen molecules. Okay. And second thing is shape of the RBC is getting changed. So shape of RBC changes. If you see shape of RBC, it is disc shape, right? Or if side view is seen, it is like a biconcave shape. But when a person is diagnosed with sickle cell anemia, or it is suffering from sickle cell anemia disease, so the RBC attains a shape of a sickle like structure, right? So this is sickle cell of RBC, right? This is RBC. You can say it is a top view, and you can say here as a side view, right? Now, it is attaining the RBC in the case of the sickle cell anemia appears as a sickle cell, right? So, in turn, what it results into is that it is obviously it is containing, or it takes less number of less hemoglobin, obviously, because of its shape. Okay. Because of its shape, it is containing less number of hemoglobin, because of which it is having or contains less number of oxygen molecules again, right? So now, because of the point mutation which is shown here, that is at the 6th position of the beta polypeptide chain of the hemoglobin, when the glutamic acid is replaced by valine, what happens? Structure of the hemoglobin changes, or the structure of the hemoglobin gets affected, because of which the less number of oxygen molecule is carried. Okay. And second thing is shape of RBCs changes, and shape is, if you see it with a top view, it is a disc shape, right? And if you see as a side view, it is biconcave. But because of the sickle cell anemia disease or the point mutation being shown, the RBC appears as a sickle cell shape. Okay. Because of which it contains less hemoglobin, and because of which it contains less number of oxygen molecule, right? So let us take, so this is the recessive, this is the dominant gene which a normal person is having. Okay. There can be a possibility, this is a person which is 50% affected with the sickle cell anemia. Okay. So 50% affected, or you can say 50% defected and 50% is normal. Okay. Now, in this case, this is 100% normal, right? Because it is not containing or not carrier. It is, this person is not a carrier of the recessive gene of the sickle cell. Okay. Now here, it can be HBS, that is both recessive gene. If a person is carrying this, it means it is 100% defected, and this kind of person do not survive. Do not survive. That's died, right? if a person is having HbA and HbS, okay, if a normal person is if a person is 50% affected and 50% is normal, then what happens? They are resistant to malarial parasite. Okay, malarial parasite. Why these are resistant to the malarial parasites? Because of the sickle shape attained by the 50% defected person, the plasmodium, which is responsible for the malaria, for causing malaria, do not penetrate, cannot penetrate actually, cannot penetrate inside this RBC. Okay, so it cannot cause malaria. So a person which is having a dominant and a recessive gene, a normal gene and a sickle cell carrier, okay, or the 50% defected or the 50% normal person are resistant to the malarial parasite. If a person is having both the dominant gene, HbA, HbA, then it is 100% normal. HbA, HbS, then it is 50% affected, 50% normal. HbS, HbS is 100% affected, and these persons are not able to survive. And these persons are getting a benefit here that they are resistant to the malarial parasite, but physical activity or the muscular activities are very much painful for them, right? Cannot be done actually. It is very problematic to show them the physical activity or thing. Okay, so now we will be revising what we have learned till now. We are studying here about the pleiotropy, right? So pleiotropy term is used when the gene is responsible for more than one character. So more than one character means here let us say that this is a chromatid, okay, and this chromatid has a gene, and this gene is responsible for various traits, right? So they are influencing various traits. So now here we have taken the example of a Drosophila. Drosophila is having the gene, and the gene which is responsible for the size of wings, okay. So now this also, the gene here which affects the size of the wings or which is responsible for the size of the wings also influences the longevity, structure of reproductive organs, bristles, and reproduction in the egg productions in Drosophila. Okay, so these affection cannot be equal. Affection rate is not equal. Right? Now we have taken the second example, that is sickle cell anemia, which is an autosomal recessive disorder, and it is occurred due to point mutation. Now we already know the hemoglobin structure. So hemoglobin has two type of polypeptide chains. Okay, so two alpha polypeptide chain, that is alpha 1, alpha 2, two beta polypeptide chain, that is beta 1 and beta 2. So in any one of the beta polypeptide chain, when point mutation is shown, point mutation in the case here, we have already seen that a normal person is having this kind of amino acid sequence. But at the 6th position, the glutamic acid is getting replaced or mutated and replaced by valine. Okay, so because of the replacement of the glutamic acid to valine, it affects the structure of the hemoglobin, and again the shape of the RBC also changes. Now, because of the change of the RBC and the hemoglobin, the less number of oxygen molecule is carried as the normal one, as the normal RBCs. So these kind of persons which are affected with the sickle cell anemia, they are resistant to malarial parasites, but they are having the problem with the physical activities or the muscular problem. Okay, so now this HbA, HbA, these two genes are normal, so 100% normal person is being shown here. Okay, now if it is having HbA, a person is having HbA and HbS, so it is 50% defected and 50% normal. Now if HbS is present, so it is 100% defected. A person is 100% defected, and because of which it did not survive. Why it is not being survived? Why it cannot be just survive with the sickle cell anemia disease with 100% of defection? Because of sickle cell anemia, or the sickle cell RBCs do not allow other organs such as, you can say, liver, lungs, heart, these are being affected. A person get heart attack, lungs failure, liver failure, okay, and a person can even get blind, because here the sickle cells are not allowing other organs to get developed, and as a result the person gets died, or death happens. Okay, so thank you for watching my video. If you like it, please like, subscribe and share my channel. Thank you.